Thank you so much. Can everyone hear me okay? Back there? Great. Good morning to all. It's very exciting to be here, and thank you for the organizer for this great opportunity to be here. I'm very excited to provide the update of what is going on at Aspen Neuroscience. And it's a great opportunity for me to talk about this transformative iPSC cell therapy, which is an autologous approach in the neuroscience space. Need to navigate this a little bit. Great. This is a company that formed on the vision to create the best characterized pluripotent derived cell medicines. And we came out of the Dr. Jean Lawrence lab at Scripps Research Institute, where 20 years of world-renowned stem cell research and the cutting-edge genomics that being created there. And Jean and the co-founder, Anders Bradley, that co-founded the company in 2018 and joined by a group of research scientists from Jean's lab and a group of patient advocates from Summit for Stem Cell. So this is a company that formed for the patient. We're highly differentiated with a disruptive iPSC platform technology. And our approach is autologous, which is the ultimate personalized medicine for every patient. So since the 2018, we have built exceptionally experienced management team and a great visionary board of the directors. So I'll elaborate some of the details that on the pipeline, our Series B funding, and also the manufacturing build out. Over the next couple slides. So in short, our vision is to be the leading autologous cell therapy company. So how do we do that? Aspen has spent years to build the foundational technologies for our platforms. So at the heart of the core is really the AI-based genomics to inform stem cell biology. Since inception, we have built and will continuously expand and augment our in-house manufacturing and quality systems. We have incredibly innovative to improve and optimize neurosurgical procedures to ensure delivering of the cell therapy in a safe and a precise manner to the specific region of the brain. So we're starting with the first indication in Parkinson's disease, and we're looking at other areas in the CNS space and very actively looking on the pipeline build out and beyond the CNS in the near future. 2022 has been a really great year and big year for the company. Now we consider ourselves to be a clinical stage Series B company. Early part of the year, we have completed all the IND enabling studies, including safety, tox, and the efficacy, and plus all the CMC activities for the first product named AMPD-001. So those are the studies required for the IND submission. We're opening our trial-ready cohort studies as we announced several months ago. So we're screening patients and ready for the initiation of the clinical trials. We have completed our significant footprint expansion by acquiring additional 22,000 square feet to continue our GMP manufacturing expansion and augmentation all the way that we believe to the phase 2B. We're thrilled to close our Series B round of $147.5 million with a group of marquee investors backing us up. We're very excited about this. So now to belabor this audience with Parkinson's disease, just to highlight, currently there is still no disease-modifying agent that on the market. Even with the current standard of the care, patients eventually develop the debilitating motor complications, which is devastating and affecting their quality of the life. The economical burden is humongous, that huge burden not just to the patient, also for the caregiver and the society, because of this degenerative disease affecting millions of the patients worldwide. So this is the Aspen approach. So through decades of the study of the disease, that we understand that that this neurodegenerative disease is caused by loss of the dopamine neuron that's residing in this part of the midbrain called substantia nigra. At the diagnostics, typically more than 50% of the dopamine neurons are already lost. So Aspen is developing a cell replacement therapy that with the goal that we deliver those cells in a safe and a precise manner to the specific region of the brain to replace the lost dopamine neurons, and potentially that for those cells to reconstruct the neural circuit that, in order to reverse some of the Parkinson's symptoms. So this is our approach. And this approach is actually backed and supported by the years of the experience of utilizing fetal tissue-derived cell therapies, which is summarized in the next slide. So this is the experience and studies started in the 1990s. Then even though there are some variable results and the outcome from the study, that we actually see some of the observations that in patients, especially the durable long-term benefit and clinical outcome to be really de-risking for our approach, which has the huge potential. The next slide actually summarizes a really high level of the overview of the current process that's being utilized to derive the first product called AMPD-001, which is an autologous iPSC-derived dopaminergic precursor cells. We started with a patient biopsy, and then followed by the dissociation to derive dermal fibroblasts, and followed by the reprogramming to iPSC clones. And some of the clones will pass the QC filtering for the subsequent differentiation to yield AMPD-001. And the folks in the audience know that making the iPSC cell products, it's not easy, it's really hard. So we integrated a very extensive QC matrix and filtering system, including pretest and the neurotest. Those are the AI-focused machine learning algorithms to predict the fate, the quality, and potentially the safety of the product throughout the manufacturing process. Here is a little bit more details into how we're actually conducting such a quality control throughout the manufacturing. So in addition to the pretest and neurotest, we also conducted the genomics tools, which are the whole genome sequencing, single nucleotide polymorphism, which is also the digital karyotyping that we utilized, and the integrated, and also the gene expression, the sequencing RNA-seq. That's throughout the process to ensure the cells are free of the tumor driver mutations or any other potential detrimental genetic alterations. We believe the approach that we're utilizing at Aspen that ensure the safety and the efficacy and replace some of the costly animal studies, such as the pretest, which is a machine learning algorithm to predict the potency of the iPSC clones. And the neurotest, again, is a machine learning algorithm to ensure the identity and determine the differentiation state of the AMPD-001 at the end of the production. So this could really potentiate the reduction of the cost and the time of the manufacturing process. So before I talk about the clinical plan, I just wanted to revisit this really landmark publication from our collaborator, Dr. Marina Amber, published last year at the Nature Biotechnology. So this is one of the longest non-clinical study in the hemiparkinsonian non-primate models to evaluate and compare and contrast autologous versus allogenic dopaminergic precursors for safety, efficacy, and durability. So as you can see from the graph at the lower left, the right corner, the blue groups are the autologous groups, which have shown the significant the advantages and the improvement in terms of motor function, which was correlated with IHC studies of the fiber outgrowth from those grafts, and also the durability of those engraftment throughout the period of the time of the evaluation. And also the PET imaging was actually shown to be very predictive of the functionality and also some of the long-term engraftment effects as well. And this is a study also revealed for the first time some of the non-motor improvements, such as the mood of the monkeys and also the self-harm being reduced. So really, really encouraging for us. So all in together, so we felt that study really validated our approach to use autologous dopamine neurons and also provided the additional the salt for fruit for further evaluation and establish the additional translational correlates and build that into our clinical plan. So this is actually the current clinical trial plan. And as I mentioned earlier, we have started our cohort, the studies, the trial-ready cohort study to start screening for the patient, which is agreed upon with the agency at the pre-IND meeting. And it was basically a way to initiate the IND filing, the submission to the agency, and to potentially accelerate the clinical development by pre-fill the pipeline for manufacturing. The primary objectives of our phase 1/2A study is to focus on the safety, tolerability, and the dose responses. So we also have built in the PET imaging, as I mentioned in Marina's paper, to be very correlative to the function, and additional clinical readouts so we can learn and capture the evidence of the biological effects and the activities for the AMPD-001 and the other informative the evidence so that we can further plan for the phase 2B clinical trial. Just very quickly to touch upon the pipeline development at Aspen Neuroscience. We're very excited for this area that, as I mentioned, so there are multiple programs that are being built out, and currently we're pursuing very active research to evaluate the multiple cell types that within the CNS, beyond Parkinson, and also beyond the CNS. We're actively looking for the partners and the collaborators to synergize this effort to potentially accelerate the development. So just to quickly to mention some of the approaches that are being integrated into the research and development at Aspen. So learned from the field over the decades of developing the cell therapy, it is important to plug in the quality of the design in the early stage of the development. So we've been utilizing the systematic analytical tools such as a factorial experimental design to generate the data that understand the inputs and the outputs, and with all the data that being further utilized to train the machine learning algorithm being established and started in Zhang's Lawrence lab. We have learned a lot how to understand, how to establish the process, and followed by the further optimization. And with the goal to really come up with the model prediction systems to ensure that not only the good quality of the product and also the product with the critical quality attributes that are desired for the specific indication can be produced through these efforts. And in parallel, we're working really hard to identify those potential technologies that would enable us to establish a fully enclosed manufacturing automation. So there are several members of the leadership that are in the audience. We're actually very excited to meet with the folks who are interested in our technology and the platform to explore the potential collaboration and the partnership to help us to accelerate these efforts. So just to kind of end my presentation on the vision again, we're the company with a vision to be the best developer of autologous iPSC-based therapy. Our approach is to bring the data sets to bear, understand it, and identify the mechanisms and the platforms to further improve and de-risk that for multiple programs and starting in Parkinson's. And then we wanted to ensure this competitive advantage that we have established at Scripps and further extended at Aspen can be strengthened through the effective and synergistic partnership and the collaboration. We believe the personalized medicine starts with you, and patients are waiting. With that, I want to thank the audience and for your patience, and see if we have the time for questions. 30 seconds.
One question?
Okay, thank you.